Thanks for returning. I hope you are coming to this video from the previous video where we began scripting the entire workflow in Bash script. Otherwise, if you have not watched that first part, I would encourage you to start from there and then you can come and continue in this video. Now, since I have my reference file and I'm sure that I have my reference file indexed, the next thing we want to do is to align our reads to the index file. And for now, I will just reorganize my file system to ensure that it's much more neat, like that. And we will now proceed to write the code for the alignment. So the code for alignment is as simple as what we've been doing. We want to simply use a for loop to iterate over some first queue reads and then perform alignment on them. Now, remember this time that we have two types of files. We have the raw first queue files that we downloaded from the sequence read archives that are stored in the data directory. We also have the first queue files that have been trimmed using first p. We want to run the alignment on the first queue files that have been trimmed by first p. Let me hash out the BWA because I don't want to run it any longer. And then write a simple for loop like for fq in. Remember, we want to iterate through the files in the first p directory. So I can just copy the first p dir like that and tell the program to do and then done. So I can continue to write my logic here. The first thing we want to do is to iterate over only the read1 files. That is why we want to tell the program to only iterate over files that end with the _1.firstq. And now from these files, we can now create our read2 files, just as we have been doing. To create the read2 files, it's pretty much simple. We just need to type in R2 and then access the read1 fq like that, and just replace the _1 with an _2. As simple as that. Now, we also want to create an output file. Now, remember that the first output of BWMM is a sum file. So the output that we want to create at this moment is a .sum file. So we can simply create it like out. And we want the file to be stored in a separate directory called the map directory. If you look at the list of dirs that I initialized here, I already have a directory here called the map dir. So I can just copy that and paste it over here and just access it like that. So the file will first of all have the file path to the map dir, and the file should be having the accession number at the end. Now here we can repeat what we've been doing. We can just use the base name. And we want the base name of the read1, that is the fq. And remember here we want a .sum file, but we don't want the .firstq. So we want to just remove the _1.firstq like that. So using the base name, we strip out the _1.firstq and then attach the .sum file extension. We can now then finally write the BWMM logic, which is just BWMM, and then put the number of threads that you want depending on your compute resources. In my case, I will just put 8. If you have more, you can include. And point the program to the reference file. And now you can just put your read1 and the read2s like that. And then we want to redirect everything to the output, which is the sum file that we created earlier. As simple as that. So our program is barely complete. Now we can just tell it to let us know that the alignment has begun. And then also ask it to let us know alignment is complete. Now what I will do is to just run the file and then see what it gives us. So it's telling me that results map directory. So this is a problem that we tried to evade earlier on, because the BWMM, just like the first P and the first QC, does not create for you the directories if they don't exist. So in our case, the map directory does not exist, so we have to create it. We can just use the mkdir as we've been doing. Just copy that and then put a -p, and then as simple as that. And then we can try running the program one more time. So basically at this moment, we have just finished aligning our reads, and we are pretty much sure that our reads are well aligned. If we inspect the results dir and then look at the map dir, you will see that we have two sum files there, one sum file for every single file that we provided earlier, just by using a simple for loop and then iterating over the files. The next thing we want to do is to convert the sum files into BAM files. Sum files are great, but for more versatility and to work in a more human readable format, we will simply convert them into BAM file. The first thing we want to do here is to hash out everything that we used to run the BWMM, because we no longer need to do that, and then write the lines of code for the sum to BAM conversion. Now here we will use the sum tools view program because it allows us to do the conversion in a much more straightforward manner. To do that, we need to iterate over the files in the map dir. So the lines of code will be as simple as what we've been doing, just writing a for loop, for example, for, let's say, some file in the map directory. Just tell the program to do and then write the done, just as we usually do. Then we can now start by creating the output that we want. We want an output that is a BAM file. And at this point, this BAM file is not sorted. So we can just create a variable called unsorted BAM. But this time we want to remove the extension here from a sum to a BAM. The easiest way to do that is to make sure that in the first line, we are accessing the sum file, because there can be other files inside that directory that are indeed not sum files. So we just want a .sum. And then simply write our code just as we've been doing. We access the sum file, and then we want to replace the sum with a BAM, just as simple as that. And then if you want, we can also create a log file that will contain all the outputs from the terminal. We can just create a log file. We want the log file to also stay in the map directory. So we can just copy and paste the line of code that we already used initially for the map directory, and simply change here from the base name. Instead of FQ, we just do a sum file, and we want to remove the _1.firstQ, replace with the .sum, and then here just write .log, as simple as that. And then the command for conversion is as simple as writing sum tools view. And then you want the program to know that you are inputting a sum file and expect a BAM file as an output. And then write the name of your input file, which in this case is a sum file, and tell the program that you want the output to be stored in a unsorted BAM file. And then redirect the standard output to the log file. Now, if you noted carefully here, we did a mistake here. Instead of putting a sum, we just need to remove the underscore, and then here simply put unsorted .bam, as simple as that. And then we want the program to let us know that it is running, as simple as that, and then say converting sum to BAM. And once it's complete, it needs to alert us that the conversion is indeed complete. As simple as that. Now, before we run this code, I want us to discuss a potential problem with how this code will run. Now, if we look at the unsorted BAM file, we are simply replacing the sum with an underscore unsorted.bam. This basically means that the program will look at any place within the file name and look at whether there is an occurrence of the name sum, and then replace that with underscore unsorted.bam. This could give you an efficient result, but in most cases, where your file structure has a sum in it in multiple areas, this can give you problems. So the easiest way to address this is to basically have another variable, for example, called base name, like so. and then simply extract the base name from the sum file. I will just copy this and then edit it a bit, just put the $sign at the beginning. And now using the base name, I can now reconstruct the unsorted.bam like this. Just copy this here and place it there. Just reformat that a little bit. And now instead of having a sum file /sum unsorted.bam, I can just remove whatever is happening there and then just copy this here. And now here at the first / I just want to have the base name like that, and then connect the base name with the underscore unsorted.bam, something like this. And then put my double quotation marks at the beginning and the end. And now, since our script is now pretty much functional, I can just save and then clear my terminal and run the program again. So it will take some time. This is something that you must get used to in bioinformatics. And the reason as to why we need the pipeline and the workflow in general is to ensure that we can dynamically handle as many files as possible without making mistakes and without wasting time. So at this point, the conversion from the sum to BAM is complete, and we can now work with the BAM file in our downstream analysis. Now, the next step is to make sure that we sort the BAM files and to index them. Why are these two important? Now, the reads in your BAM files are in the order in which the sequencing platform gave it to you. But when we want to do the variant calls as well as when we do the visualizations, we basically want to maintain the order that the reads exist in within the genome. And that is why we need to sort the files. Now, we need to index the files to enable us to easily and randomly access the BAM files without needing to read the entire file. So the first thing I will do here is to just comment out all these sections, and then just write a for loop to both sort and index the BAM files. So in this time, we want to write a for loop that targets the unsorted BAM files. And at this time, we want to iterate over the files in the map directory. So I will just copy this. And here, I no longer want to target the sum file. I want to target the unsorted.bam files, just like that. And remember, the name that you use here should be consistent with the name that we used here. So it should be underscore unsorted.bam, like that. And we can now write done and write the logics of the code here. We want to create the output of the sorting and the indexing steps. And we basically want to retain the BAM files, but we want to change the name from unsorted BAM files to sorted BAM files. So we can create a variable called sorted BAM and target the unsorted BAM files, and just replace this unsorted.bam. So it should be unsorted.bam. And so we can just replace this here with sorted.bam. Just as simple as that. And then the logic is just some tools. First of all, start by sorting. So some tools sort. And we target the unsorted BAM file as our input. And then tell it that we want the output to be the sorted BAM. Now we want to combine this with the sum tools index so that we run them both concurrently. And to do that, we just put a double ampersand. This will make sure that the program on the left runs first and then the program on the right runs second. Now this will be different from piping, as we will see in our next step. So we can just write the sum tools index. What we want to index is the sorted BAM file. So we can just copy this like that and redirect everything to a log file. We have not created this variable, but we can just put log and then create the log file here. just like that. And then we can recreate the path to the log file. We can just copy this and paste it there. Still everything in the BAM directory, base name, but targeting the base name of the unsorted BAM file this time round. And what we want to remove is underscore sorted.bam, and just put the .log at the end. So we need to verify that the code is correctly scripted. And if you note in our naming system, you can first of all note that here we needed to put just underscore unsorted.bam and not the underscore, because that is how the naming convention is here. The other thing we also want to correct is, instead of just putting here a log, we want a log underscore bam. And the final thing we want to make sure is that we write the for loop correctly. So it will be for unbam in that directory. And we can tell the program to let us know that the step is running. So it's sorting and indexing BAM files. And then also let the program alert us that the step is finished. As simple as that. And then now that the code is fairly well scripted, we can simply rerun the program again and see what it gives us. So you can see that we did not catch one of the problems. It's directly connecting the sample name with the sorted.bam. So we can simply correct that. What we can do is just terminate this and just correct that as soon as possible. Here we want an underscore sorted.bam. So everything can now run correctly. We can just rerun the entire program again. So you can clearly see that it's correctly giving us the underscore sorted.bam files. And that will be important in our next steps. So I will just close the directory so that it looks a little bit neater, but you don't need to do that. So the program has sorted our BAM files, and it has also indexed those BAM files. Now, if we hover over the map directory, we can clearly see that we have sorted BAM files. Other than the sorted BAM files, we also have additional files here called the sorted.bam.pai. That basically tells you that the files have indeed been indexed. The next step here is to call the variants, but before calling the variants, we need to run an Mpileup of the bases. Now, let us first of all write the code or script for Mpileup and the variant call, and then explain what the Mpileup does. So I will start by commenting out this section. The lines of code for the Mpileup is just as simple, and we've been doing this all throughout the scripting process. We simply need to create a for loop, but this time we want to target the BAM files, and not just BAM files, but the sorted BAM files. So we can create a variable like BAM, and then we want to target the files in the BAM directory. So we can just copy this here. Done. And remember, this time we want underscore sorted.bam. So we just remove that, as simple as that. The next thing to create is the output file. So remember that the output file of the variant call process is a variant call file. So we can simply create a variable like a variant call file and then point this to a file within the variants directory. So remember here, I initialized a directory called the variants directory. So I can just copy this and then say that I want the file to first of all have the path to the variants directory, just by doing that. And then say that I want the file to have the file extension and the file name that goes alongside the accession number. So basically just do base name as we've been doing. And then we want to do the base name of the sorted BAM. Like so. And what we want to remove here is this underscore sorted BAM and replace it with a .vcf. And finally write the command for Mpileup and the command for variant call. So we want to do Mpileup using the vcf tools Mpileup, and then we want to tell the program that we expect our output to be a variant call file, and also inform it that we want to supply it with the reference file and provide a path to the ref file. And then remember that the input to the BCF tools Mpileup is the sorted BAM file. And here is where now some level of triviality comes in. We want to pipe the output from this program to a second program. So we use the pipe function of bash script. So we pipe the output generated from the BCF tools Mpileup to a separate program called BCF tools call. Now, in terms of calling the variants, we can call the variants using multiple, I would say, programs or algorithms. In our case, we want to call the multi-allelic variant caller. So we called it MV. And then tell the program that we want the output to be a variant call file and point it to the path of the output, which is the VCF file that we created earlier. So here at the BCF tools call, we can either put -mv, or in some cases, there's also another caller called -c. The difference between -c and -mv is that -c is a consensus caller. -m here is just a multi-allelic caller. Now, multi-allelic caller is much more accurate and gives you multi-allelic variants. The consensus caller is less accurate, but it can sometimes work in cases where you are conscious much more about the time and the speed of your program running. But in most cases, you will want to call the multi-allelic caller. Now, this program is well scripted. We can verify that it runs. We can tell the program to let us know that it is running the Mpileup and variant calling, and also let it inform us that the process is complete. So let's review the functions of the Mpileup. So what the Mpileup does is pretty much simple. It examines all your align reads and then creates a summary of the bases present at each position in the reference genome. Now, for each genomic coordinate, it basically gives you the reference base there. It then gives you the number of reads covering that position, and then it gives you the actual bases called in the reads at that specific position. Other than that, it will also give you things like the quality scores of those base calls. Now, given that we are much more confident about the script, we can just run this code. I can just clear my terminal and run the file again. So here it is telling me that it cannot find the indexed file compressed with gzip, and then it tells me that please use gzip. So this can be a common problem that you may experience in your day-to-day analysis, and that basically tells you that you need to be very careful with your reference file. In our case, the reference file was a .gz zipped. So I am not exactly sure about how this file was zipped, but clearly the program does not like this file. So what I will do here is just copy this file, copy its path, and then use the gunzip to unzip that file like that. And then now, instead of writing here .fna.gz, I will just remove the .gz now to just work with the .fna, and then rerun the program one more time. And it throws to us another problem that it cannot find the variants directory. So this is a problem that we've been trying to avoid all along. So we can just make that directory, like just copy this and then use the mkdir-p, then just create the directory, as simple as that, and rerun the program one more time. Hopefully it doesn't have any more errors. So the program is happy. It is running the Mpileup and the variant call. And at the end of the day, we will have our variant call file. Now, the program has finished calling the variants, and we can be sure that if we access the variants directory, we can now access our variant call files. So we can just do this, and we can clearly see that there are two variant call files here. Now, for these tutorials or for these videos, I will not be focusing in depth into the details of these VCF files, but for the sake of those who are new to bioinformatics, I just want to gloss over the contents of these files. So let's open one file here, for example, that file. This is just normally how messy the variant call file is. It starts by these hashtags here that basically gives you some level of metadata of the variant calls. And then if you scroll down, you get much more important information about the variant call process. Now, what I want to focus on is here. So here you get much more meaningful information. For example, you get the command that was used to call the variants. But not only that, you also get information about the chromosome position, chromosome ID, the reference base at that chromosome position. You also get information about the alternate base at that position, and then the quality of the variant call. Now, if we move to the right side of the file, you will find a very important information here. And this piece of information will be important to filter out bad variants. The DP here tells you about the read depth. It basically informs you about how many reads support this alternate base that was called by your variant caller. A read depth of 1 just shows that only one read supports that variant, and a read depth of 4 means 4 reads supports that variant. In most cases, when filtering out, you will want a variant to be high quality if at least 20 reads support that variant. We have another annotation here called the SGB. You can clearly see it here. SGB simply stands for segregation-based score. So it's a penalty that is applied when the variant looks too good due to perhaps some things like under segregation. It's quite much more specific to the BCF tools. We have another annotation here called the MQOF. What MQOF basically means is the mapping quality of 0. It tells you how many reads have a mapping quality of 0. A score of 0 basically tells you 0 reads had a mapping quality of 0. That's a little bit much more informative. We have another annotation called AC. We can see it somewhere here. AC is an annotation for allele count. It tells you how many alternate alleles were observed in that position. Now, the final thing I want to highlight is the annotation called MQ. So MQ is annotation, for example, like this. It tells you the average mapping quality at this position. So an MQ of at least 31 is fairly good enough. Here we have MQ going as up as 60, which is fairly good enough. During variant filtering, we will want to use some of this information to filter out our variants. And now let's go back to our main script to filter out bad variants. I will just close this and make sure the file structure here is way much more organized, like that. And then also clear my terminal. I will want to comment out these sections. So the command for filtering the variants, we just need to write a for loop for VCF in the variants directory. And we want files that end with a .vcf. Do done. And we can now write the logics of our command. So we want an output, and the output we want is a filtered VCF file. So we can create a variable that holds the filtered file. Now we can just obtain this by pointing the file to the path of the variants directory. So we can just copy this and paste it over there, and then put a // and then use the base name to strip the file path of the VCF file. And remember now what we want to strip out is the .vcf and attach the .filtered VCF file annotation. This is much more easy to read. Now we can write the filtering process, and here we will use the BCF tools filter functionality. So we want to tell the BCF tools that our output should be a variant call file, and then we want to specify the filtering formula. So we just do a -i and then put double quotation marks and then write your filtering formula or filtering methods here. So we want to start by looking at the quality of the variants. For example, we have the qual. So we want a qual of greater than 20. Now, if you want, there are current publications that shows that a quality of 20 gives you much more confident variants. Now we want to combine this with a read depth of greater than 10. And at this point, I just want us to stop there. But if you want much more stringent variant filtering, then you can repeat this process and look at those annotations that we discussed earlier, and then pick on your most favorable annotation. So we want to point the BCF tools filter to an output file, and the output file will be the VCF filtered file. And then finally give it the input as the VCF file. We want the program to let us know that it is running the process. And also let it inform us that the process is complete. So we want to counter check our script to make sure that it is correctly scripted. And you can see here that we made a few mistakes. For example, this should not be here. It should be there. And also make sure that you put the asterisk there. And now, since we are much more sure about our program, we can rerun the file again. It's a very straightforward and a very quick process. But how do you verify that the variant filtering happened? Now, one thing you can do is to basically look at your variants directory, and you can see that there are two files here. There is the .filter.vcf file, and then there is the file that we obtained in our earlier analysis. But really, that doesn't tell you whether the filtering process happened. The other thing you can do is to just look at the size of the file. So you can note that the unfiltered variant call file could be larger than the filtered variant call file. The most sure way to do that is to literally count the number of variants in both files. For example, we can count the number of variants using grep. Simply write grep -vc. So the C here means to count. So it will count all the entries, and the V here means that it will exclude the entries that we will define next. For example, here I am defining the entry to be hashtags. So I want the program to not consider entries that have got hashtags before them. And now I will just apply the path to the variant call file. I will start with the unfiltered variant call file, copy the full path, and paste it over there. And you can see that the number of variants here are 145,770. I can repeat the same process for the filtered variant call file. Just copy the full path, just delete this. And run it, and you can see here that we have 14,562 variants. So it basically tells us that the program did a massive variant filtering, and most likely we have high quality variants, but you are never sure about this until you look at the statistics and you do additional statistical analysis, validation, as well as visualization. So I will just clear my terminal and just say that at this point, the entire pipeline is well developed, is well finished. If we delete everything in our directories and remove all the hashouts in our code and just run the program again, the running will be so systematic. Start with the topmost command, going all the way to the last command. Now, in the next video, we will discuss how to do this in Snakemake. So I want to thank you for listening. And if you want, you can stick around for the next video.